Today I'm going to talk about epigenetic regulation of cardiomyocyte phenotypes. And the overall goal of my talk today is to demonstrate that epigenetic mechanisms may play a causative role in the development of a disease phenotype. So first of all, I'd like to start off by defining epigenetics. So as defined by Dr. David Alice, an epigenetic phenomenon is a switch-like change in phenotype that is heritable but does not involve DNA mutation. So if you look at the classic dogma of genetics, that states that basically DNA controls everything. So DNA begets RNA, begets protein, and the way that you can inherit phenotypes based on classic dogma principle is that you can have mutations at the level of the DNA which are then heritable. Epigenetics adds to the classic dogma in that DNA begets RNA, begets protein, and the protein and RNA can feed back upon the DNA and alter expression patterns of the DNA, and this altered DNA with a new phenotype is subsequently heritable. So we know that these mechanisms are very important during development. So cellular identity during development is established in the nucleus by way of stable epigenetic modifiers that control the expression and repression of genes without altering the underlying DNA sequence. So we know that embryonic stem cells, shown here, are essentially epigenetically naive, and as these cells start to differentiate, they obtain epigenetic marks which start to restrict their expression patterns until ultimately they become differentiated cells. And we know from work in development that these epigenetic marks are fairly stable, and we also know that they define gene expression patterns and ultimately define the phenotype. So in my lab, we seek to determine whether epigenetic changes in differentiated cells could provide a mechanistic explanation for the alteration in gene expression profiles that results in disease. And I think that adult cardiac myocytes are an interesting cell type to study because they are largely non-dividing and long-lived cells. Thus, by studying epigenetic mechanisms in adult myocytes, we can explore the importance of maintaining the epigenome over time. And so this is my sort of little diagram about how epigenetics may contribute to disease. And so I call it a stepwise accumulation of persistent changes. So here's the stable, normal epigenetic state. And over time, you may have subtle alterations in the epigenome, which then cause persistent stable changes. And the accumulation of these persistent stable changes may ultimately result in or predispose to a disease phenotype. So in my lab, the epigenetic marks that we study are histone tail modifications, particularly histone methylation. This diagram shows you DNA wrapped around a protein spool, which is called the histone, and these histones have tails that protrude out around the DNA, and these tails are amenable to post-translational modifications. So for example, these tails can be methylated, and these methylation marks can help facilitate active gene expression, or they can obtain methylation marks that will be important for repressing the neighboring genes. So if you look here and you look at different types of genes, different genes will have different epigenetic profiles. So if you look at the 5' regulatory region of silence genes, they will usually have an enrichment of H3K27 methylation marks. However, you will find a paucity of this mark at the 5' regulatory region of active genes. In comparison, H3K4 trimethylation marks, which is what I study in the lab, you generally will see a paucity of these marks at the 5' regulatory region of silence genes. However, you'll see a significant enrichment at the 5' regulatory region of active genes. So the way that we have studied these marks is we have used a protein called PTIP. PTIP is part of a histone methyltransferase complex and PTIP interacts with the enzymes that are important for imparting H3K4 trimethylation marks that are associated with actively expressed genes. And the way that PTIP knows which histones to modify are that it interacts with DNA binding proteins or transcription factors which provide the specificity on the DNA. After the DNA binding proteins bind, they bind with PTIP, which subsequently recruits the histone methyltransferase complex, which ultimately puts H3K4 trimethylation marks at the neighboring histone, which renders that gene actively expressed. So in my lab, we use a lot of tamoxifen-inducible cardiac-specific deletion models using a FLOX-PTIP allele and an alpha-MHC Mercury-MER model. We were able to show that when we inject tamoxifen in these mice, we pretty much completely delete PTIP protein in adult cardiac myocytes. In order to determine whether or not this complex is important for maintaining gene expression profiles, we perform gene expression arrays to identify potential target genes. Of the many target genes that we identified, some that were particularly interesting were potassium channel genes. So we showed that KCNE1 was significantly decreased when PTIP was deleted. And importantly, we showed that KCND3 or KV4.3 and KCHIP2, which make up an important part of the transient outward potassium current, were also significantly decreased when PTIP was deleted. So in order to show that those genes, particularly KCHIP2, was specifically regulated by the PTIP complex, we performed CHIP assays, which allow you to associate histone methylation marks and chromatin-binding proteins to certain areas of DNA. And basically, by using CHIP assays, we are able to show that when PTIP is present, the histone methyltransferase complex is present, we're able to maintain the H3K4 trimethylation marks, and we're subsequently able to maintain KCHIP2 gene expression. In the absence of PTIP, we aren't able to localize the histone methyltransferase complex to the 5' regulatory region of KCHIP2. We aren't able to maintain the H3K4 trimethylation marks, and as a result, we have a decrease in KCHIP2 gene expression. Looking at this mouse in more in depth, since we knew we had alterations in the potassium channel, particularly the transient outward current, we did surface EKGs, which showed in the PTIP null mice they had a significant ST depression, and measuring individual action potentials, we were able to show, this shows you a normal mouse action potential, which is strongly regulated by potent transient outward current, and the PTIP null mice, we have a very prolonged action potential duration. We went on to look at the transient outward current specifically in these myocytes, and this shown here shows you that there is a significant decrease in the transient outward current in PTIP null mice as compared to mice that have PTIP intact. By giving these mice caffeine and isoproterenol, we were also able to show that this does indeed induce a disease phenotype in the form of a susceptibility to increased ventricular premature beats. The question that we've been looking at more recently is what role does this methyltransferase complex play in the cardiac adaptation to stress? So to study this, we have set up a transverse aortic constriction model whereby we put a 27-gauge needle across the transverse aorta, tie it off, and pull the needle out, resulting in pressure overload-induced hypertrophy. We have a sham group and a TAC group, and we use PTIP plus and PTIP null mice. And at 1 week, we do an echo, and at 2 weeks, we do an echo, and then we harvest the tissue and look for pathology and in the future gene expression profiles. And as what we found is that in the absence of PTIP, shown here, at 2 weeks, we get a dilated hypertrophic heart in comparison to the mice that have PTIP intact, where we develop the expected concentric hypertrophic response. If we look at the function of these two different groups, 1 week and 2 week after TAC, we can see that in the PTIP null group, here's baseline cardiac function. 1 week after aortic constriction, we have a significant and persistent drop in the ejection fraction, which we don't have when PTIP is still present. So in summary, H3K4 trimethylation marks regulate the expression of potassium channel genes, including KCHIP2, and inability to maintain these marks results in an attenuated ITO, prolonged action potential duration, and a depressed ST segment by EKG, as well as an increased susceptibility to ISO and caffeine-induced ventricular beats. It also seems that PTIP is necessary for the development of concentric hypertrophy in a pressure overload model, and inability to maintain PTIP results in cardiac dilatation and failure. In conclusion, an inability to maintain epigenetic marks in a fully differentiated cell type results in changes in gene expression and the development of a disease-like phenotype. Epigenetic instability thus can be a causative mechanism of disease. So in the future, I'm very interested in sort of focusing on this PTIP histone methyltransferase complex. I would like to figure out what other genes are regulated by it, and I think the easiest way to do this at this point is to employ ChIP-seq technology. I would like to figure out the mechanism whereby the PTIP complex regulates this cardiac response to stress and why I'm getting a dilated cardiomyopathy. I also have some other mouse models that I'm using where we can delete epigenetic proteins that are important for maintaining repressive marks, and also we can delete chromatin remodeling enzymes that are important for unwinding the DNA and the transcription of active genes. I would like to acknowledge my mentor at the University of Michigan, Greg Dressler. I would also like to thank Dr. Halife, who, without his help, none of these studies would have come to fruition. And I would just like to thank the organizers again. This is a really great experience.